But in case of fruit breeding crops, where the handling of larger population is very limited and we need high resource to maintain that population, this method is very like cumbersome. Actually, you can't use that bulk breeding method in the fruit crops, vegetable crops, where we have to we need a larger resources to handle the larger population. That's why this method is not that much followed in the fruits and other vegetable crops, but it is followed still now also like modified form of bulk breeding we are following. Actually, we're not following exactly bulk breeding, but we are following modified form of bulk breeding that we will discuss in next slides. And another thing is, another disadvantage is, as I told, breeder involvement is very less. Up to F8 there is no involvement of breeder, just it's seed multiplication, just you are harvesting, and you're not doing any individual plants like individual progenitors, you're just bulking then advancing. The breeder attention is very low, means the breeder can't exert his intelligence here. That's why this method is less favored by breeders, because they have limited time, they had to develop a variety in a very short duration of time. That's why there are all disadvantages will uh, leads to the less adaptability of this breeding method in improving the variety.
Hello, sir.
Hello, sorry, I was, yeah. This is about a bulk breeding method. Actually, that's why it's because of this disadvantages, so nobody is adopting this method for the improvement, varietal improvement. But modified forms are majorly used, like single seed descent method, mass pedigree method. There are several modifications to the bulk breeding method. Like it's also a modification for pedigree method, or it is called as mass pedigree, and another one is single seed descent. Single seed descent is mostly used method for development of reels. Many breeders are actually adopting this method to advancing generation and developing the real population. We have discussed what is real population, what is the advantage of real population in mapping. This method is single seed method is very much used. And another modification of the pedigree, it's actually modification of the pedigree, and it's also modification of a bulk. It is called as mass pedigree. Actually, it was proposed by Harrington. For example, you are using a pedigree method to develop a resistance for some diseases. Sorry, some diseases. But in that case, you are not getting any diseases because of the environmental condition is not that much favoring for development of the disease. So that doing a plant to progenitors and evaluating, it's no use actually, because when you don't have a particular environment, selection environment, then you are unable to select the better plant. Then growing a plant to progenitors, it's a waste of time. That's why scientists thought that why can't we maintain this population in a bulk up whenever the favorable condition observes, you just grow plant to progenitors and start your selection procedure. That's why instead of going a plant to progenitor selection without a selection environment, just go for a bulk breeding method until you're getting a favorable environment where you can exert a selection in a better way. Okay, that's what after getting a better environment, you start your pedigree selection. That's why it is clubbing of your both pedigree and the bulk breeding method to attain the better selection environment to get the better individual, to do better screening. Okay, that's what the mass pedigree. It is actually proposed by Harrington in 1934. Understood what is mass pedigree? It just the environmental conditions are not favorable, and it's very difficult to distinguish the plant or a progenies of individual plant selection or a plant progeny in a particular area. Means you have to harvest in a bulk, in a then you just advance in a bulk only until the favorable condition will prevail for the efficient pedigree selection. This is what mass pedigree method. Understood what is mass pedigree method of breeding and bulk breeding method? Any doubts?
No,
Just
sir.
I am revising. No doubts. Okay. Just I am revising the basic things because it's there in the syllabus. Okay. Then what is single seed method? We have everyone know. Actually proposed by Gordon in 1939. How? It is same as like your bulk breeding method. Here just a difference is you are advancing only single seed from each plant and you are advancing that seed. Every rest other everything is same. You are doing you are not doing selection at F2. You are just simply advancing the generation. Instead of advancing all the seeds from all the plant, you just take one seed and you grow in the next year. Just take one seed from this generation, grow in the next year. The purpose of this breeding method is like for example, your development of reel, you should, there is no need of they'll if selection will if you do selection means you can't maintain entire segregating population till up to F8, and you just maintain all segregants till F8 so that they will reach homozygosity. And so, for example, if some quantitative rate, you may get you can able to identify some plant which having all the better for this quantitative trait rather than in F2, F8 you may get that plant. In F2, because of it's governed by large number of genes, particular trait, governed by large number of genes, that this important segregant you may not get in F2. But in F8 you are going to get. Then with minimum resource, we can handle this entire segregant until F8. And this is the one of the biggest advantage over bulk. Means minimum resource is required because you are advancing only single seed from plant. The if you are if the population is 100 here, in F8 also the population will be 100 only. Means there is no increase in the population size. We can easily handle the entire population. Even we can use a greenhouse condition because we have very limited population. You can put in greenhouse condition and you are not doing any selection here. Means, for example, you are not doing, for example, you are doing any selection for agronomic trait, you should be grown under the normal field condition. Okay, but you are not doing any selection and the limited population size that you can grow in any greenhouse condition or any protected cultivation so that we can take multi-season like without any seasonal dependence. You just grow how many number of generation you want. You just grow like that many generation in a single year. That's what the beauty of this single seed descent method. You can advance rapid advancement of the genotypes, segregating population, and maintaining the intactness of the entire segregating population without any extra burden. And we can exert a better selection for the quantitative rate in the F8 because of the and remember there is no environmental variation like there is no natural selection in the single seed descent method because we are growing a single plant whatever it means a kind some kind of favorability you are doing because if you're selecting seed you're not obviously you're selecting better seed only. Okay, that's why you just think that there is no natural selection in the single seed method, and also you just keep on advancing into F8 without any selection for the desirable trait like agronomic traits, and you can do entire breeding procedure in a protected cultivation. That you can save time, resources. That's why these are the different advantages of the single seed descent. But what is the disadvantage of single seed descent method is the identity of your F2 cannot be maintained because it will reach to homozygote. But somewhat like some breeders say, you may not get the identity of F2. The if you do F2 again, you select that plant. You keep on selecting means the better segregant within that favorable plant will be very high. But it is a confusing point, like still in a debate. But as of now, you just remember, you're selecting a single seed from each plant. You are advancing at F8 without any selection, and we require very less number of times. Means we can grow several generation, rapid generation turnover, because we can use off-season environments like pollution greenhouses, and we can have a minimum resource to maintain a small population. And we can able to, this method is widely used to develop a real population or to develop a rapid homozygous population that can be served as inbreed for hybrid development. These are the several advantages of the single seed descent. Understood what is single seed descent method? Any doubts? And who given the Gordon was given. Okay. Then backrest method. Actually, backrest method is very important method. Discussed in detail about backrest method while discussing about marker assisted backrest selection. Okay, that's why I'm going very split. And these all breeding method I have discussed in detail while discussing about marker assisted selection. Okay, that's why I'm going very fast. And backrest method is as given by Harlan and Pope. And please remember the scientist for each and every breeding method. Scientist and year also very important. Okay. And Harlan Pope has actually discovered this method, backrest method. It's just a procedure. It mainly depends on either you can transfer a single gene or a multiple gene, which may be dominance or which is maybe a recessive. It may be governed by a quantitative inheritance, whatever it may be. You are just transferring one gene, like some genes, and you are putting into some genotype without altering any character of that genotype. Okay, we're just correcting some defect of that genotype. That is called as backrest method. And here, what is the different character of the trait under question or trait under transfer? Normally, we used to get if it is a dominant method, like if it dominantly the particular gene is gone by dominant allele, a particular allele, if the desirable allele is dominant, very easily we can transfer. If it is gone by recessive, we will discuss. We need an other step to get to it. You have to make that express in a generation, then you have to do selection. We will discuss that. But if it is a dominant allele, it is easily you can transfer. But if it is recessive allele, we need some extra generation of selfing to identify that recessive homozygote. Then, if it is controlled by a quantitative character, we need another one method, like we need some modification of backrest method. For example, you are doing a multi-trait transfer. In detail, I have discussed how to do a multi-trait transfer, how to do a different genes from different donors into a single variety. We have discussed in detail. Then, if you're transferring a dominant allele, what is it? For example, you see in this example, I think is for transferring a rust-resistant gene from a donor parent to recurrent parent. And actually, in this figure, it is given vice versa. Actually, normally, should use recurrent parent as a female. In only while transferring the male sterility, we have to use recurrent parent as a male. But in other cases, we generally use recurrent parent as a female, and always female should be written this side. Okay, that point you should keep in mind. But here some mistake, I think. Okay, then you're transferring this particular gene to this background. Just you cross it and you develop an F1. You backcross to the recurrent parent. Then you generate BC1 and generate BC2, BC3, BC4, BC5 until you may get, it depends on you. For example, these like backrest progenies, how much degree of similarity you want the type of the recurrent parent until you have to do backrushes. You know that what is many times asked in the ARF, ASR exam, ASRB exam, why we need to do backrush especially means to recovery of recurrent parent genome. Just remember recovery of the recurrent parent genome we have discussed in detail. That's I'm not going in detail. Then after doing backrust, once you think that okay, you thought that okay, you attain the genotype's maximum similarity to the recurrent parent and also plus the gene of interest. Then you keep on selfing to get that gene of interest to get homozygous condition. When you recurrent parent, maximum recovery of recurrent parent plus homozygosity of the donor gene, then you just go for the evaluation trials, replicated multiplication trials, then you go for seed multiplication and distribution. This is about a simple procedure for backrest breeding if it is controlled by a dominant allele. But if it is controlled by a recessive allele, you need an extra 1 year. For example, in BC1, if it is controlled by recessively, the resistance this RR susceptible, this heterozygote will be susceptible only because small r will not express in the your heterozygous condition. That's why you just self it, then you observe, the evaluation for your desirable trait in the segregating population, F2 population, BCF2 population. Then you cross your desired individual to recurrent parent. Then you again generate a back cross. Then you again self. Then again select. Then again you cross. Then you just need an extra generation of selfing after each back cross. Then it doubles your actually your breeding procedure, actually breeding time to transfer whenever you transfer recessively. That's why we have found an alternative of MABB. Even the trait is dominant, even the trait is recessive, irrespective of that, you can just go for BC2, BC3, then you just keep on selfing and release. Means half of the time will be saved. Within 6 to 7 years, we can able to release the variety if you're using an MABB. Okay. Can anybody tell what's the steps in MABB? We have discussed in detail. What are the different steps we follow in MABB?
Foreground selection, background selection, recombinant selection.
Exactly. Okay. Foreground selection, background selection, and recombinant selection. We are exerting these different selection based on the markers. Then we can stop at BC3. We can get maximum recovery of recurrent parent genome within this generation only. Based on marker screening, we identify that plant which having maximum recurrent parent genome recovery. Then you keep on selfing. Then you release variety. Means you are stopping your back crosses. Then you start this procedure in this year only. Then yield trials. Then you will release it directly. Means you are saving half of the time. That's why MABB is mostly used than the any conventional backcross in the nowadays. Actually, in every lab they are going. For example, at least some maize, rice, wheat, all the crops which are having a good reference genome and good marker systems, they're all going for MABB like tomato, chili, all those crops. Those some orphans crops like some legumes, still we are following conventional methods, but most of they are shifted to marker state backcross breeding selection that we have discussed in detail. Okay. For example, if the trait is governed by any doubts?
Yes, sir.
Yeah.
Sir, in backcross selection, as we go for advancing generations, backcross after backcross, by selecting the desired character along with most of the recurrent parent genome earlier itself, we can reduce the duration in traditional backcross itself, no, sir? Is it possible,
Well,
sir?
some
In
but
traditional,
no, that cannot. See, when you do backcross only, the recovery of the recurrent parent genome will be very high.
yeah,
If
yes,
you
sir,
keep on
that's what I'm
selfing, if for example I understood your doubt, in first generation of backcrossing only, after single backcross, that can also be done. Okay, that can also be done. Okay, after single backcross, you keep on selfing. That may take some more years to recover the recurrent parent genome. Okay, after single backcross, you keep on selfing up to F9, F10, you may get, but the chances is very less. You need to handle a large population to maintain that turn. Okay, in F2, we need to maintain the table I have forgotten actually. When you compare F2 and backcross, the recurrent parent genome recovery in both the condition you are going to get. But you need to handle a large population to identify this plant, which see in backcross, if you are doing a backcross, how many genotypes you are going to get? One is homozygote, another one is heterozygous. But how many genotypes you are going to get here? 4 genotypes you are going to get. Okay, one heterozygote and 2 homozygote. Means you had to see 4 individuals to select one gene, but you had to observe only 2 genotypes to select one gene. Which one is better?
Backcross.
Backcross. Okay. Because when you do backcrossing, you get only two genotypes. One is homozygote, one is heterozygote, 1:1 ratio. But here you are getting 1:2:1 ratio, means 4 individuals you are going to get. But here we are going to get only 2 individuals. Okay. That's why you need to handle the less number of population. But and again you are getting more recovery of the recurrent parents because we're continuously crossing with the parent you what you want. Okay. That's why backcross is always beneficial than the F2. It's not like that in F2 you can't get this type of plant. You can get, but the handling and the number of generation you instead of out of 1,000 you may get 20 plants which are exactly look like your recurrent parent genome. But here out of 100 maybe 75% will be your recurrent parent genome. That's only the difference. That is a simple genetics. Okay? Understood?
Yes, sir.
Forgot actually one table was there. It's very beautifully explained. Like how we can what is the difference of genetics you're going to get when you're doing selfing and when you do a backcrossing. Okay. If the trait is quantitative inheritance, the trait is governed by a quantitative character, then what we can do means because the trait is governed by many number of genes, so that to getting a better segregant will may take some more time. That's why instead of doing selection in BC like F1, what you are doing, you are doing selection in F1 and crossing to any donor. But instead of doing that, you just keep on selfing like you as you told, like BCF3, then select the better plant, then you do back crossing again. It may take some more time, but it will be the better approach as per the previous reports. Okay, understood? What is the difference between the normal qualitative trait and quantitative trait, the backcross breeding procedure?
Yes, sir.
Yeah. Then again, if you want to do the hybrid, one ruling hybrid was there, but it is susceptible. For example, if it is lack of your protein content, you want to introduce some FLORI2 gene and OPAQ2 gene into the hybrid. Then what backcross? What is the mode of backcross cream we have to use to improve this hybrid?
Sir, we have to improve the parents first, I think, sir. Defect
Yeah,
correction in
exactly.
parents.
You had to do, for example, parent 1 and parent 2. You to do backcrossing to improve the parent 1, and you have to do backcrossing to improve the parent 2. Then again you make combination of this improved inbred line. Then you identify the genes, then better heterotic combination. Then you do go for release. Okay, it is very tedious task. Okay, here you can just improve only one parent, like you're just putting a gene from here to here, and then you do. But here in hybrid, it's not like that. You have to improve the both the parents, the desirable genes. Then you keep on improving. Then you cross the improved line. Then wherever you're getting the better heterosis, that combination you have to select. Then you go for yield trials. That's what the difference between the backcross for self-pointed crop and the hybrids, improving the hybrids. Okay, understood? What is the difference between them and this? These are the method, and backcross can be used for both the breed, like all the irrespective of the mode of pollination, we can use cross pollinated, self pollinated, often cross pollinated. It's not like that; it is only used in the self pollinated crops. Then, if you want to transfer a more than one character, you can use simultaneous transfer. Either you can use separate backcrosses and you can merge. Then you can use stepwise transfer. We have discussed in detail. Refer my MABP class for this. Then, what are the different application of backcross breeding? Okay, this year one question was asked. How we can develop near isogenic lines? Either through backcross, either through mutation, either through any methods? Backcross, mutation, either through CRISPR-Cas9, either through gene substitution. In any ways, we can develop a near isogenic lines. Okay. Then another method, like we can use backcross for development of near isogenic lines. And the only method to transfer the different cytoplasm to desirable lines, because of backcross only now we can able to develop CGMI system in whatever the cultivar we want. We can incorporate our male sterile genes due to the availability of this backcross breeding scheme. Then we can intervalated transfer of simply inherited traits that we have discussed. Then we can use it for germplasm conservation. Nowadays, ABQTL approach is very famous. We have discussed what is ABQTL approach. It's very muchly used in the pre-breeding. Backcross is very predominant breeding method in doing pre-breeding. Means you are, instead of bringing the wild variety into the breeding pool, first you backcross with the important varieties, develop near isogenic lines of these donor genomes. Then you use that nils in the actual breeding program. That's what is called as pre-breeding. We will discuss what is pre-breeding, who given it in detail in coming classes. And one of the major problem in conventional backcross is the linkage tract. If the desirable trait is linked with an undesirable, it's very difficult to transfer through conventional backcross breeding. That's why one of the important modification in MABB, which selection is, that is recombinant selection, that is majorly used to transfer the gene of interest by avoiding the linkage tract. Okay, please refer MABB class how we can use a recombinant selection to avoid the linkage tract to transfer the desirable trait. Okay, these were the different application of backcross breeding, and everything which are discussing the basic seems like a basic, but many times it asked in the SRP exam. Okay, then, and apart from these methods, there another one special method is called as diallele selective method. It's not majorly used, but the scheme was hypothesized to use this method to improve the broadening the genetic base of the self-pollinated crops. Actually, we are using pedigree method. It will end up with developing pure line. If you do backcross method, it will end up in the developing of pure line like homozygous line. If you do bulk breeding, it is end up in developing homozygous line. Because all the breeding method is aimed to develop a homozygous line, it means that you are narrowing the genetic base. During the course of time from 1900 to 1970, the most of the self-pollinated crops, the breeding population, having very narrowed genetic base because of we are aiming to develop a pure line with a uniform genome, like a uniform parental genome. That's what it hindering the productivity and stagnation of the yields, and also it become more vulnerable for the disease and pests. That's why scientists thought to introduce, if you want to introduce um, variability in this self-pollinated population, how we can bring, because it's naturally it's inbreeder, but it's self-pollinated. Then how to, we can't make them to cross-pollinated. We can make, but it needs a very time and laborious. As a scientist thought, why can we hypothesize a breeding method which improves and increases the broadening of the genetic base of the self-pollinated crops? Then that scheme was designed to attain the recombinant desirable gene from multiple parents. Means in a breeding population, let me just enlarge this. Sorry, this figure is not that much good. Say, for example, the parent you are using, like the initial population, you just develop an F1, then allowing crossing among this F1, then it's like a diallele. Like in diallele, what we will do? You are allowing F1 to recombine within all possible cross combination. Then, if you want to develop pure line, you keep on selfing this F1, then develop a pure line. But if you want to do, again include some parents, then you allow for diallele mating, then you select the better individuals, then again you keep on crossing, then again it will serve as a source population for next generation. If you want to develop a pure line, select some better line, allow continuous selfing, then you develop a pure. Then again here, this and the cycle will follow here. This is what diallele selective mating means. You are making all possible cross combination in every parental generation, and you are selecting the best individual. Then keep on advancing. You are developing a pure line. In one hand, we are developing a pure line, and in another other hand, we are making an heterogeneous population and allowing the recombination to happen. Means you are increasing the genetic base and also without compromising the development of pure life. You are doing two jobs. One is you are doing keep on selfing. You are developing a pure line, and also you are allowing for crossing, and you are developing a source population with having broader genetic base, because you are allowing crossing means it's allowing the more recombination to form. Then again it serves as a population for next generation selfie. Then you just do F2, F3, F4 and you develop your line. Then allowing anyhow you are making diallele crosses, then again you do selection here and keep on going this. Okay, this is what a diallele selective mating means. You are making a diallele crosses, then you are again selecting, then again you are making diallele, then you are selecting. Means you are doing two jobs. Okay, understood? What is diallele mating scheme? And it was given by Jensen. Many times it asked in the exam. Please remember who given it. And what is the main motto of diallele selective mating is to improve to broadening the genetic base of the self-pointed cross. And what are the main advantages of this method is you can broaden the genetic base, you can use multiple parents and you can allow recombination between them. Then when you allow recombination, what will happen? We can break some undesirable linkages so that we can allow linkage drag also. Then and we can opportunity of having introducing germplasm at any stage of program. We can introduce here, here and here, anywhere you can use new parents. Then so that we can further broaden your genetic base. Means we can also fulfill short-term goals. Means you're allowing recombination and you're broadening genetic base, short-term goal. And also your long-term goal, like if you go for F6 and you can develop pure lines. Okay, this is what the advantages of the dial selective mating. Then what are the disadvantages? Means you are doing a diallele selecting means the seed setting is very, very low. I think those who have done crossing in self-pointed curve, they know that like crossing itself is difficult and getting the setted seeds is very, very difficult. Then we need, whenever all the stages you have to maintain a diallele population means we require a large number of seeds. That is a major limitation in this method. And that's why nobody is following this method. But you just prepare, you just know that what is diallele selective mating, who coined this term, and normally why it is used. Okay, then still it is under process. Nobody has practiced. Still, it needs to be rectified with the many problems, and some character with low heritability can't able to select. But there are some disadvantages also there. You just read in the BDC book. Okay, these are the points which are for diallele selective mating. The another method is we can use recurrent selection for self-pointed also, and it is given by Palmer. But still, it is not that much used. But you just remember you can use recurrent selection also. You can use diallele selective mating method also, and you can use mutation breeding also, polyplate breeding also in self-pointed crops, along with what I have discussed in this class. Any doubts? Okay. That I am completing a breeding for self-pointed crops. I completed a hybrid breeding. Same like whatever we have points we have discussed. Same thing applies here. I am discussing rice crop tomorrow so that we will discuss what are the male sterility systems are there, how we can develop hybrid in rice. We will discuss that in rice crop. Then population improvement method I have discussed in detail. And mutation breeding I will discuss. And backcross, pedigree, mass, introduction I have discussed. And marker selection also I have discussed. Then genomic selection and gene editing will go. Okay, this is the end of breeding for self-pointed crops. Tomorrow I will discuss rice. Okay, what is the rice? What are the breeding methods we are following in rice? The important rice genome project. How the rice become a model crop for a genomics. Okay, we discussed in detail. Any doubts from up to here? No doubts. Jayanth, any doubts? You will join newly.
No, sir. No, no. Sir.
Okay. And follow all like watch my all recording videos. I think tomorrow you'll get approval. You watch all my older recorded videos so that you can catch what I have dealt until now. Okay.
Hi, sir.
Bye. Yeah. If no doubts means, shall I close the class? Cut.
No doubt, sir.
Okay. Bye. Good night. Good night. Good night, everyone. Bye.